Hello everyone. Today we are going to learn about a process which is known as protein synthesis. This is nothing but the production of protein take place inside our body, as protein plays very vital role inside our body. Protein synthesis is a three-step procedure in which transcription, translation and translocation occurs. So we'll be understanding protein synthesis in a stepwise manner. Let's talk about transcription. To understand transcription, we're going to have DNA, and for DNA we need to zoom in inside the cell. So this is the cell. Inside cell we have nucleus, and the outside portion of the nucleus is known to be the cytoplasm. The periphery of the cell is known to be the plasma membrane or cell membrane. If we zoom inside the nucleus, we are going to have the DNA. DNA is our genetic material that is going to be transferred from parents to offspring in each generation. Now, when I talk about DNA, DNA is a double stranded structure and it is going to help us to make the protein. How it is going to help us to make the protein? So for that we need to understand the transcription procedure. For that we are zoomed in inside the nucleus. So we have our DNA. DNA is a DNA is made up of two strands. For this procedure we are just going to use one strand of the DNA. So I have decoded the DNA to make the sequence. As we can see the sequence, it is made up of 4 bases, that is A, T, G and C. This is one of the DNA strand. This DNA strand is going to be transcribed to make mRNA. So in this whole protein synthesis procedure, we are going to use two types of RNA. One is mRNA, which is used in transcription procedure. So this DNA is being transcribed to form mRNA, which is messenger RNA. This is done by an enzyme which is known as RNA polymerase. This is an enzyme. What it is going to do is, it is going to make the sequence as AUGC. So in DNA we have ATGC and in RNA we have AUGC. So the thymine is being replaced by uracil. So far mRNA is there with us. Inside the nucleus, DNA is being transcribed to form mRNA because DNA is very bulky and it cannot transfer to the cytoplasm. So we have mRNA, this is a short fragment and it can be traveled to the cytoplasm. So the further translation procedure is going to enter inside the cytoplasm. So we are in the cytoplasm, we have the mRNA sequence that we have in front of us. So the base pair now are going to make a group of 3 and that is known as codon. Now for each and every codon we are going to have an anticodon which is going to be brought by the tRNA. This is the second type of RNA being utilized over here. This type of RNA will be bringing along with it two things. One is the amino acid and the other thing is anticodon structure. This anticodon structure will be complementary to that of codon. So here we are going to have U, that is complementing A, then we have A which is complementing with U, and then we have C which is complementing with G. This is the anticodon structure of this codon. Each tRNA will come with the anticodon structure as per the codon and one amino acid. As per the anticodon, amino acid is going to be linked with the tRNA. tRNA, which is you can see over here, this is tRNA, that is the transferred RNA. The main use of this tRNA is to bring the anticodon and amino acid as per the codon. This is a hairpin like structure, and what the amino acids are going to do, the amino acids are going to link together like this. We are going to have 20 amino acids all together will come to form one protein. So what are proteins? Proteins are nothing but they are made up of amino acids. Amino acids are the building block of the proteins. So like that, each tRNA will come to make the anticodon structure and we are going to have the sequence. The next thing is the ribosome. As you can see, there is a pink color thing over here, which is nothing but the ribosome. Now, why the ribosomes are important? Ribosomes are known to be the protein factory of the cell. Okay, so they are really important because they are the one which are going to make the protein. So they are the protein factory inside the cell. So without ribosome, the proteins are not going to be prepared, and that is one of the reason ribosome cannot travel to the nucleus and DNA cannot travel to the cytoplasm, because both are bulky, and because of that we have mRNA been introduced over here, which is going to carry the message of the DNA to the cytoplasm where the ribosomes are there, and there the proteins are going to be produced. So like this, the proteins will come. Now this is the second protein, that this is the second amino acid that will come, and like this we are going to have all the amino acid making up the chain. And this is how, when the 20 amino acids will connect with each other to form the protein. Now this is the overall sequence that I have made over here. One is DNA being transcribed to form mRNA, that is messenger RNA. This procedure is happening inside the nucleus. Then the mRNA travels to the cytoplasm, and inside the cytoplasm the mRNA will help us to make the protein. And how it is going to help us? With the help of tRNA. So tRNA will be bringing the anticodon and amino acid with them, and that will make up the protein. And in this, ribosome is going to play a very important role, because that is the protein factory of the cell. This is the theoretical thing that I have written over here. So if you want, you can pause the video and you can see what is being written theoretically, so that you can make your answers more attractive. This is also the same thing. Now I am just remaining with the translocation. Translocation is nothing but the procedure which involves the ribosome. So when I say ribosome, ribosome is the protein factory, and when this is the mRNA, let's say, when the mRNA enters the cytoplasm, the protein factory, that the ribosome, attached to the mRNA. Now when it gets attached and the tRNA is coming and getting attached to the codon, what exactly it is going to do is, it will just move. The ribosome will keep on moving from one end to the other end as per the triple codon. That means the codons that are there on the sequence of the mRNA. That is nothing but known as the translocation. Why it's moving? It is moving to make the protein, and therefore ribosome is considered to be the protein factory of the cell. So this completes with the transcription, translation and the translocation procedure. So I repeat the overall structure over here. DNA to mRNA is transcription, and if I talk about the translation, that is basically mRNA to protein. And in between, when there is involvement of the ribosome, we have the translocation procedure as well. So this completes with the protein synthesis procedure. I hope it was useful for you all. If you like the video, please hit the like button and please subscribe to our channel. Thank you.